This is USMLE QA, USMLE material presented in a question and answer format structured similar to the first aid for the USMLE Step 1 book.
Question: what is fluorescence in situ hybridization? Answer: fluorescence in situ hybridization is a technique used for specific localization of genes and direct visualization of chromosomal anomalies, where a fluorescent DNA or RNA probe binds to specific gene or other site of interest on chromosomes. Question: what is the use of fluorescence in situ hybridization? Answer: fluorescence in situ hybridization is used for specific localization of genes and direct visualization of chromosomal anomalies. Question: what are some examples of chromosomal anomalies that can be detected using fluorescence in situ hybridization? Answer: some examples of chromosomal anomalies that can be detected using fluorescence in situ hybridization include microdeletion, translocation, and duplication. Question: what is a microdeletion and how is it detected using fluorescence in situ hybridization? Answer: a microdeletion is a chromosomal anomaly where there is no fluorescence on a chromosome compared to fluorescence at the same locus on the second copy of that chromosome. It can be detected using fluorescence in situ hybridization. Question: what is a translocation and how is it detected using fluorescence in situ hybridization? Answer: a translocation is a chromosomal anomaly where the fluorescent signal that corresponds to one chromosome is found in a different chromosome. It can be detected using fluorescence in situ hybridization. Question: what is a duplication and how is it detected using fluorescence in situ hybridization? Answer: a duplication is a chromosomal anomaly where a second copy of a chromosome, resulting in a trisomy or tetrasomy. It can be detected using fluorescence in situ hybridization. Question: how does fluorescence in situ hybridization work? Answer: fluorescence in situ hybridization works by using a fluorescent DNA or RNA probe that binds to a specific gene or other site of interest on chromosomes, allowing for the specific localization of genes and direct visualization of chromosomal anomalies. Question: what are some of the advantages of using fluorescence in situ hybridization? Answer: some of the advantages of using fluorescence in situ hybridization include its high resolution, high specificity, and ability to detect both small and large chromosomal abnormalities. Question: is fluorescence in situ hybridization widely used in the medical field? Answer: yes, fluorescence in situ hybridization is widely used in the medical field for the diagnosis of chromosomal abnormalities and genetic disorders such as Down syndrome, Turner syndrome, and others. Question: can fluorescence in situ hybridization be used to detect cancer? Answer: yes, fluorescence in situ hybridization can be used to detect cancer by identifying genetic changes in cells. It can be used to detect specific genetic changes in cancer cells, and it can also be used to monitor treatment response. Question: is fluorescence in situ hybridization a new technology? Answer: no, fluorescence in situ hybridization is not a new technology. It was first developed in the 1980s and has been widely used in the medical field since then. Question: can fluorescence in situ hybridization be used to detect specific sequences or genes? Answer: yes, fluorescence in situ hybridization can be used to detect specific sequences or genes by using probes that bind to specific sequences on the chromosomes. This makes it possible to identify specific genetic changes or mutations in cells. Question: is fluorescence in situ hybridization a safe technique? Answer: yes, fluorescence in situ hybridization is considered a safe technique. It is non-invasive and does not require any type of radiation exposure. Question: how can fluorescence in situ hybridization be used in research? Answer: fluorescence in situ hybridization can be used in research to study the structure, function, and organization of chromosomes and genes. It can also be used to study the role of specific genes in disease and to identify new targets for drug development. Question: are there any limitations of fluorescence in situ hybridization? Answer: some of the limitations of fluorescence in situ hybridization include that it can be time-consuming, it requires specialized equipment, and it can be difficult to interpret the results. Additionally, the technique is not always able to detect all chromosomal abnormalities. Question: what is the difference between fluorescence in situ hybridization and other genetic techniques? Answer: fluorescence in situ hybridization is a technique that allows for the specific localization of genes and direct visualization of chromosomal anomalies, while other genetic techniques such as PCR, sequencing, and microarray are used to detect and measure genetic variations or mutations.
Thank you for watching our Q&A format video. We hope this helped you learn, review, and retain knowledge needed for the USMLEs. For more information on this topic, check out the link in the description. Now, if you want to take your USMLE prep to the next level, be sure to check out all our courses. which are structured to follow along with the first aid for the USMLE Step 1 book. You can find us at usmleqa.com. And if you're watching this on YouTube, be sure to like and subscribe.